And I am looking forward to seeing and hearing about the research that you did this summer. What was it, 6 weeks, 8 weeks?
8 weeks.
8 weeks. And I just want to commend you and let you know that you are shining stars and you have some wonderful people who are very much interested and invested in your well-being. They took the time to assist you this summer, and I want to also congratulate them. So without further ado, again, I'm proud of you, and I'm going to yield and let the program begin.
Thank you, Dr. Lang. So, and then Dr. Carter, so if you say something as the chair of the department.
Yeah, as chair of department, it is just great honor. You've just bestowed a great honor upon us that we still have research, even though we had to kind of be on lockdown, but your advisors and program faculty saw fit for our research to continue no matter what situation. And you guys were really good to oblige them. So we're really excited about that because you learn so much by doing, by hands-on, as I'm sure you guys realized from your work that you did this summer. So I, for one, am excited to see what this research is all about. One, for it being from the department, and the other from being from our department students. So we're just, and we're so proud of you. So we're just ready to hear what all you learned and all the challenges actually that you had in your learning. That's also a very, very important part of it as well. Okay.
Thank you.
Thank
then
you,
I want to ask Dr. Samiono, and he is the PI of this grant, and and also I am the co-PI of this grant. So I just want to request Dr. Samiono talk about something, the program, how it goes, all 8 weeks, and actually we worked, I mean the 8 weeks, and our students worked very hard, I mean 8 weeks to get some excellent results, what actually they will present. So first I want to request Dr. Samiono to say something.
Yeah, thank you. Good morning, everyone. Yeah, first of all, I'm really concerned about the program because, we don't have a face-to-face meeting, something like that. So, and then I have an idea to put the what do you call it? Put the program in Canvas. So I started the program from Canvas. So everything I ran the program through the Canvas. So we set up the Canvas as like our campus. Okay, like you can say like virtual campus or virtual lab or something like that. Okay? So some of the students really they start from zero. I can say they start from zero. Like they didn't know how to program in Python. They didn't know how to. They don't have idea about what is mathematical modeling, what is the something like that. Okay. So I guide them through the Canvas. I put some material there so they can learn about the Python. They can learn about the research and mathematical modeling, especially in cancer research. I mean, mathematical modeling for the cancer growth or the modeling for the cancer treatment. Okay. Yeah. Finally, I think they can deal with the problem. Some of them like live in southern Texas. As you remember, recently we have like Hannah hurricane. So 3 of them was affected by the hurricane. So we had to postpone the presentation. Okay. So finally we can make the presentation today. Okay. Yeah, I think that's all that I would like to convey to you. Oh yeah, one more thing that also they learn about latex. Latex is like mathematical software to type mathematical formula or mathematical manuscripts and like that. So they also learn about this. So in the final report, they will write the report in LaTeX. Yeah. Yeah, I think that's all that I can contribute to you. Okay.
Okay. Thank you, Dr. Samiono.
Yeah,
That actually all for hard work, and also students really work hard during this the 8 weeks program, and today they are also excited, so they will present their work. And you know, the work actually is a mathematical biology, so what actually we include like the biological work, how we can model mathematically to get the more precise, indication. So one of our target was like mathematical modeling of our, for killing the cancer. So, actually my work and all over like the last few years, our students also worked on the cancer cell, different cancer cell, mainly the breast cancer, and also they worked with some of the nanoparticles. Dr. Carter are involved mostly in this work, and also different other cancers. Okay, so the finally, like in this summer, because we could not go to the lab, but it was really, really hard work to get, all the data to like the mathematically model. So I think that actually real, a difficult task. So, but we have done this, and our student now will present, and they will present alphabetically from the last name, right? So please, when our student will present their research, please just mute yourself and then hear. And after the presentation, if you have any question, there is a question answer section, so you can ask any question. So please just mute your mic or microphone and hear and also know just what actually our student has done over the last 8 weeks. And I told you they really, really work hard and every day. So please be patient and hear what actually they learn during this summer work. So first I would like to welcome to present Jordan Bradley. So please share your slide, Jordan, and present your research.
Yes, sir. Oh, Jesus.
Yeah, so open the first one.
Okay. Good morning, everybody. My name is Jordan Bradley, and I will be speaking today on the mathematical modeling of the effects of a crude onion extract on MCF7 human breast cancer cells. So today we'll have our introduction, which will go over how can mathematics help cure cancer? What is mathematical modeling? The purpose of our research? What is Python, as well as some equation descriptions that we used. We'll also go over, briefly touch on our previous research as it is connected to the research that we did this summer. Our materials, our methods. We'll talk about our results. We'll discuss our results and then have a conclusion. And then at the end, we'll have a Q&A. So our introduction. In our abstract, we talk about how in this research, our objective was to mathematically model our previous research, named studying the effects of an onion extract with a focus on quercetins on MCF7 human breast cancer cells. We used 6 different mathematical equations to determine that the Mendelssohn model was the was the mathematical model that best represents our growth curve, and with a final SSE of 366,697.865, which we'll discuss all of this later. This is just a brief overview. While the linear model was best represented our water control treatment with a final SSE of 7.601 to the negative 10th power. And lastly, the best model that represented our 33 micrograms per milliliter treatment was the Bertalanffy model with the final SSE objective of 6.754 to the negative 17th power. So our introduction. In this research, our objective was to mathematically model our previous research named Studying the Effects of a Crude Onion Extract with a focus of quercetin's effect on MCF7 human breast cancer cells, in which MCF7 human breast cancer cells were used to try to determine how effective a crude onion extract is in treating MCF7 human breast cancer cells. During our previous research, it was determined that with more research, a crude onion extract could possibly be a viable treatment option for cancer. And throughout our current research, we use 6 different mathematical equations, those being the exponential equation, the Mendelssohn equation, the logistic, linear, surface, and Bertalanffy equations that represent different types of tumor growth that allows us to determine which equation best represents our data. We used an optimization method known as the POW method to show which can solve our equations without using derivatives, to find which type of tumor growth best represents our data. The effectiveness of our equations is determined by what's called the final SFC objective. And we use the final SFC objective to determine the effectiveness of the modeling equations by comparing them. And the lower the number returned, the better the equation represents our data. In this article, we will be examining the effectiveness of 3 types of previously received results: a growth curve, a water treatment or water control, and a 33 micrograms per microliter treatment. So how can effectiveness help cure cancer? We can answer this question since computational experiments that test various cases can be performed quickly and with no risk to living persons. Additionally, these mathematical and computational tools should even allow us to tailor treatments to individual patients, something that is not commonly done in medicine today. So what is mathematical modeling? A mathematical model is a description of a system using mathematical concepts and language. Mathematical models are used in the natural sciences, such as biology and chemistry, and engineering disciplines, such as computer science, as well as social sciences, such as political science. A model may help to explain a system and to study the effects of different components and to make predictions about behavior. So what is the purpose of this research? The purpose of this research is to use Python to mathematically model our previous research named Studying the Effects of a Crude Onion Extract on MCF7 human breast cancer cells. The reason we're doing this is to bring a better understanding of our previously received. So what is Python? Python is a general purpose and high-level programming language. We can use Python for developing GUI or graphical user interface applications such as websites and web applications. Also, Python is a high-level programming language which allows you to focus on core functionality of the application by taking care of common programming tasks. Python is an excellent programming language for scientists and can be used at many levels, from simple scripts for quick calculations and big programs by implementing complex data models. So here we're going to start going over our mathematical equations, the ones that we use, just to give a brief description of how these models would describe how a tumor grows. So the exponential equation would say in the early stages of a tumor, cells divide regularly, creating 2 daughter cells each time. A natural description of the early stages of cancer growth is thus the exponential method, where growth is proportional to the population. The proportionality excuse me, I'm sorry. The proportionality constant A is the growth rate of the tumor. This model is often used in early analysis of tumor growth curves and appears to work quite well at predicting early growth. However, it is known to fail at latter stages when angiogenesis and nutrient depletion begin to play a role. The Mendelssohn model is a generalization of the exponential growth model, which was introduced by Mendelssohn. In this model, growth is proportional to some power known as B of the population. The logistic model was created by Pierre Francis Verhulst in 1838. This model describes the growth of a population that is limited by the carrying capacity of B. The logistic equation assumes that the growth rate decreases linearly with the size until it equals 0 at the carrying capacity. The linear model assumes initial exponential growth that changes the growth that is constant 1 over time. In our formulation of the model, the initial exponential growth rate is given by A over B, and the latter constant growth is A. The model was used in early research to analyze growth of cancer cell colonies. The surface model assumes only a thin layer of cells at the surface of the tumor are dividing, while the cells inside the solid tumor do not reproduce. They are meiotically inactive. Our formulation again assumes exponential growth at early times, with the surface growth is taking over at longer time. And lastly, the Bertalanffy model. The Bertalanffy equation was created by Ludwig Bertalanffy as a model for organism growth. This model assumes that growth occurs proportionally to surface area, but that there is also a decrease in tumor volume due to cell death. This model was shown to provide the best description of human tumor growth. Okay, now to briefly touch on our previous research. So here's the abstract. It says that onions are a very important part of diets for many people, being consumed in countries all over the world. Onions are also used in many natural remedies for all types of illnesses and have been scientifically proven to help alleviate many illnesses, like cancer. During this study, a crude onion extract, known as COE, was used to find the effects on human breast cancer cells, on a line of human breast cancer cells called MCF7 cells. The anti-carcinogenic effects of 2 different crude onion extracts were examined on MCF7 human breast cancer cells. During this study, the two different time periods were also used to find how effective the crude onion extract was over a 12-hour treatment period and a 3-day treatment period. In our current research, we studied the 3-day treatment period. And the results concluded that crude onion extracts would be a remedial path in inhibiting the progression of human cancer. And so here's some of the data. Here's some of the data that we'll be graphing. And so this is the 6-day growth curve, which represents the pattern of cells, or the growth pattern of cells when untreated, and when left untreated and grow in optimal conditions, which was 35 degrees Celsius at 5% carbon dioxide air content, and RPM in a medium called RPMI 1640, and 10% fetal bovine serum with 1% antibiotic. Here is our second. Here's the second treatment data that we'll be analyzing. And so in this one, we'll only analyze the water control, or the red line, and the 33 micrograms per milliliter treatment, which is the light or lavender color line. And so this table represents the data obtained when cells were treated over a 3-day period with crude onion extract at 4 different concentrations with 2 different controls, one being water and one being the medium previously discussed. So onto the materials. So in our current research, we only used 3 things. We used Python, which was our coding language, Spider, which was a program used to code, and in our previously discussed mathematical modeling equations. And so our methods, we utilize the different mathematical models in Python. I apologize for there being a copy, but here are the equations, here are the equations. So our exponential equation was y equals alpha times y. Our Mendelssohn equation was y equals alpha times y to the beta power. Our logistic equation was y equals alpha times y times 1 minus y over beta. Our linear equation was y equals alpha times y over y plus beta. Our surface model equation was y equals alpha times y over y plus beta to the 1/3 power. And our Bertalanffy equation was alpha times y to the 2/3 power minus beta times y. And here are our results. So starting from the left, from the top left, I apologize for these not being labeled, but so starting from the top left, going to the right, it is the exponential model, our exponential model, our Mendelssohn model, our logistic model, our linear model, our surface model, and our Bertalanffy model. And so this is our growth curve data. And as you can see, looking at, so the red line shows, the closer the red line is to the black line, which is our actual data, and the red line is our optimized data, or the data that whenever we put it into our equation, we receive that data back, and it is optimized to show which type of data fits our model the best. So as you can see, looking at our graphs, our exponential model fits the best. While looking at our data, at the actual numbers received, and trying to determine from the final SSC objectives, our Mendelssohn model represented our data the best. Now moving on to the water treatment. So again, starting from the top left, going to the bottom right, it goes exponential, Mendelssohn, logarithmic, linear, surface, and Bertalanffy. And so looking at it, our Bertalanffy model fits the data the best. However, looking at our numbers, the linear model seems to, due to the final SSC objectives, the linear model is shown to best represent our data. Next, we go into our 33 micrograms per milliliter treatment. And here again, we can see that our bottom right, our Bertalanffy model represents our data the best. And looking at our numbers, the Bertalanffy model actually does represent our model the best this time. So to discuss, looking at our data, we can determine the effectiveness of our model in representing our data. One way that we can do this is how well we observe the data is by one way that we can observe our data, or how well this data is represented is by examining the way that the optimized data fits our originally observed data. However, we get a closer look at this through our optimized numbers. By looking at our optimized data, we can observe that the Mendelssohn model serves to best represent our growth curve data. This can be explained by the fact that the Mendelssohn model represents tumor growth that's proportional to the population, and we can see how the growth curve serves to give a sense of the way our cells would grow when unaffected and in optimal growing conditions. The model that represented our growth curve data the worst was the logistic model, and this could be explained by the purpose of the logistic model, which is to describe the growth of a population that is limited by the carrying capacity. showing that on one hand, one model shows as the population of the tumor grows in proportion, while on the other hand, another model shows that the population growth is limited by the carrying capacity. Nextly, looking at our water treatment data, we can see that the linear model represents our data the best. We can rationalize this by reading that the linear model is used to represent data where growth is constant over time, moving from exponential to a more constant form of linear growth. This explains how the linear model best represents our data, because we use the water treatment as a control to express that the water used in our extract, because our extract, our crude onion extract, was a water-based extract, would not affect the growth of our cells. Hence, our cells were growing constantly and had no major hindrance to their growth. The model to most poorly represent our data, our water control data, was the Bertalanffy model, and this can be reasoned by the fact that it not only assumes that cell growth is proportional to surface area, but also that there is a decrease in tumor volume due to cell death. This would make the Bertalanffy model a poor representative of our water control, because we can assume that there is little to no cell death occurring. And this assumption is supported by our data in the KP4 column, which was a column of optimized data, which represents a decline in cell number, because the optimized data is, and since the optimized data is positive, meaning that there's a lack of cell growth. Lastly, we will discuss our 100 I apologize, it's meant to say 33 micrograms per milliliter treatment. In this treatment, we find that the Bertalanffy model is the model that best represents our data. We can conclude that this model best represents our data because, as previously discussed, the Bertalanffy model best represents data where growth is proportional to surface area, as well as where there is a decrease in tumor due to cell death. And since these cells are actually being treated, it makes sense that the best fitting model would be the Bertalanffy model. However, the model that represented this data the worst was the exponential model. The possible rationale behind this could be that the exponential model is known to fail latter stages of cell growth when nutrient depletion is a possible factor for cell growth. Our conclusions. Throughout this research article, multiple tumor growth models are used to analyze the treatment growth and control treatments for our previous research. We used 6 different tumor growth models, in which all represented a different type of tumor growth. When these models were applied to our previous data, it could be observed that when the models best represented by a type of data, when representing our growth curve, the best model was the Mendelssohn model, while when representing our water control treatment, the best model was the linear model. Whereas when representing our 33 micrograms per milliliter, the Bertalanffy model was the best. Moving forward, we will attempt to further this research by using mathematical modeling to determine which treatment has a greater effect on THP1 human monocytic leukemia cells. A pure quercetin extract, which was the bioactive molecule being examined in our previous research, or the crude onion extract, which was being examined in our previous research. Here are our references. Anybody have any questions?
Okay, thank you, Jordan. Yeah, it's a nice presentation. So you use the only one extract, right? That actually the continuation of the previous research, and also you presented in the annual biomedical research conference, right? So
yes, sir.
it is the more, excellent, what is called the experience for you to just model your previous data to the mathematical modeling to get the more precise, identification that which model actually more effective, right?
Yes, sir.
Yeah, great. So faculty, students, staff. So if you have any question, please just ask Jordan. Any question?
Okay. Yes, this was a fantastic presentation. I really enjoyed it. It was real good. I was able really to follow in terms of your discussion and your conclusion. I really like to see that. I'm sure you learned a lot from this work, looking at your Python and all of the mathematical modeling. It was just really interesting. How do you see using this to extract what I know you showed a couple of things, and you showed some extraction from your work in terms of mathematical modeling, but what makes this different from just what you did this summer and what you're doing now, what you did today? I mean, what you did this past summer, what you did the summer before, and what you're doing this last summer.
So the difference between this summer and last summer, and really what our work was it helps to show how the tumor grows due to our treatment, and how the tumor, how our treatments actually affected the tumor's growth. So as I was saying, with my 33 micrograms per milliliter, it shows that the cell death was, well, when compared to other equations. So I believe the surface model, which showed that cells only grew on the surface, and not that tumor cells only grew on the surface and not inside. Whereas in the Bertalanffy model, it shows that cells grow all throughout the tumor, and they grow proportional. However, there's also cell death, and that's what the main difference was. We could determine how our cells actually grew.
And that is specific for each cell line, as pertaining to your particular personal treatment. So if you used a different cell line, perhaps that would be a little different.
Yes, ma'am. That's what our future research, we plan to determine how not only how our bioactive molecule affects the cell differently, but also if it affects the cell in ways like how it grows.
So if you turned it around a little bit and could you do the mathematical treatment without actually doing the cell treatment, the cell modeling, the actual wet lab?
I don't believe that we could, because in order to have the data to mathematically model, we have to, in order to mathematically model the data, we must first have the wet lab data.
One more question. I'm gonna let somebody else go ahead and ask. Now, you did the growth curve, the water, and the treatment. Okay, what was in just the growth curve alone? I'm just asking. What was just the growth curve alone? What was that?
The growth curve was our representation of how our cells grew over a 6-day period.
I understand that, but it was just, did you just use plain media?
Oh yes, no. We use plain 1640 RPMI.
Okay.
Media only, right?
Yes.
I have some more, but I'll let somebody else ask. I saw you had a question in your chat. You might want to look at that.
Okay. How does your research tie into treatments on the market today based on onion extract?
Okay, yeah.
That's a great question. So I'm not 100% sure how our research ties into treatments on the market today based on onion extract, because the treatments on the market today aren't just onion. It's more of specifically refined molecules. So here at Jarvis, what we did was we used a crude extract. So basically it was just blended onions and water to see how the onion would affect it. However, and treatments on the market today would be like actual pure what's the word for actual pure elements in
pure crystalline.
Right.
yes.
What is carrying capacity?
Okay, the carrying capacity is how much life a certain space can provide. So, or how the carrying capacity is how much how many organisms a single environment could carry. So, for an example, if we were to take the world today, some scientists estimate that the carrying capacity for our planet is 9 billion, and we're at 7 billion right now, quickly approaching our carrying capacity.
Excellent. Excellent presentation. Thank you.
Thank you. And so,
Jordan, any question? Or you can also put your question in the chat box so we can see.
okay, does your research tie into treatment? Oh, he said thank you. It's no problem. Thank you. And so now I'll give my for my acknowledgments. I want to thank God for providing this wonderful research opportunity, the NSF for providing the grant that allowed us to do this research. I want to thank Dr. Semiano for all the hard work he's done, and Dr. Bouillon for working hard to make sure that there was a research opportunity this summer. I also want to thank my fellow researchers who helped to make this a very enriching experience. And lastly, I want to thank the people, everybody who came before me to help pave the way for me today.
Thank you, Jordan.
Thank you all.
So our next presenter is Andrew. Jarvis, right? Andrew, are you here? Next presenter is Andrew, or?
Yes, yes, Dr. Yeah, yes.
Yeah. So Andrew, you can just share your screen and then you can just present. And you know that Andrew is mathematics major, right? And he is the real mathematician. So we can see what actually the Andrew has done over the last 8 weeks. He also used some of like the previous research on the cancer cell, and his research actually he used like the crude ginger extract. That ginger extract actually we did the last summer. Who did it, right? Yeah, Sharti great, right?
Yes.
Yeah. So one of our previous summer scholar. So Enro just use the data to do the mathematical modeling. So Enro, please just present your research.
Okay, well, good afternoon, everyone. I'm Andrew Salas, and I'm here to present what I accomplished over this 8-week program. The title of my presentation is Using Mathematical Modeling to Study the Anti-Cancer Effects of Crude Ginger Extract on MCF7 Human Breast Cancer Cells. And I'd like to acknowledge right now the program which allowed me to do this, and then just experience this over the summer and just help me like learn and grow and just broaden my horizons, which is Jarvis summer undergraduate research experience in computation and mathematical biology. And also want to thank my mentors, which is Dr. Bouillon and Dr. Samino, who helped us a lot throughout this time. And also the the team members who are all a part of this with me. And I'm the presenter. So today we will begin by going over what I'll be presenting today, which is the abstract, the introduction, the materials and the methods, the mathematical models, the results and conclusions. Abstract. From when time began, humans have been utilizing Earth's natural resources for own benefit. However, it was not until recent years when there were significant discoveries in the biomedical field opening new doors to other uses of resources. Scientists have revealed several fruits, spices, natural herbs, some of which have shown a dual uses, such as hidden medicines and or treatments. Ginger, which is Zingiber officiale, contains shogo and ginger ale, which has been shown to have an anti-proliferative effect on several tumor cell lines. This study focuses on the utilization of mathematical models to study the anti-cancer effects of crude ginger extract on the human MCF breast cancer cells. And this is based on Mrs. Gregg's experiments that she did last summer, which I believe. So, what is cancer? Cancer is a disease caused by uncontrolled division of abnormal cells in a particular body. Despite significant advances in medicine, breast cancer continues to be a chronic medical problem worldwide, being the most common type of cancer in women. According to the National Breast Cancer Foundation, 1 in every 8 women in the United States will develop breast cancer in her lifetime, and 1 in every 39 women will die from it. The MCF7 is a commonly used breast cancer cell line which was proposed in 1973 by Herbert Soule. So the introduction. In mathematical biology, mathematical modeling can identify the space of knowledge and logical inconsistencies and can help guide us to where to keep looking and where not to. Mathematical models can provide insight and knowledge over cancer cells, such as it helps biologists and mathematicians to predict and learn new behaviors and adaptations of cancer cells, starting with a theoretical formulation of a problem can unleash numerous opportunities of testing hypotheses and simulating scenarios and therapeutic interventions in a completely safe way. Furthermore, mathematical modeling is a method that is not only utilized within breast cancer cells, but can also be utilized in colon, prostate, lung, and liver cancer. Also, it shows us other benefits that natural herbs such as ginger is beneficial for and other problems of the body, such as inflammation, decreasing blood sugar levels, and nausea, just to name a few. As analyzing the study, we're shown that ginger inhibits the growth of cells and reduces the low protein. It is also showed us that the proliferation of MCF7 cells treated with ginger extract was markedly decreased to 35% compared to the control at the concentration at 0.11 milligrams per milliliter. This study which was previously in the research that Mrs. Greggs did, and in that study, it was predicted that the crude extract of ginger would have anti-cancer effects on MCF7 breast cancer cells. However, in this study, we will be testing how ordinary differential equations, which is the mathematical modeling, can predict and highlight the best models to utilize for certain scenarios. And we will see how the data presented will allow us to determine the best fit and optimized model and would be the Bertolin. So in order to get my data, Mrs. Griggs had to do certain procedures and methods, and which this is just what she did. The cell culture cells are obtained in an negative 80 degree freezer. When ready for procedure, they are thawed in an incubator for 5 minutes. Added RPM at 1640 at 10% FPS, a 1% antibiotic medium that has been warmed in the water bath for 10 minutes into a 15 milliliter centrifuge tube with cells in centrifuge at 1,000 RPMs for 5 minutes. When done, aspirate and added 9 milliliters of media to the centrifuge tube with the cells and mix it. When done, put the 1 milliliters of cells in the 75 cm2 culture flask and 9 milliliters of the media and incubate. The cells are to be culture for 3 days in the incubator to become 100% confluent. When that is done, you're able to play every 24 hours for 6 days. The cells were retrieved out of the incubator. The medium was aspirated and rinsed with the PBS. Transynthesization occurred. Cells were continued using the highermost cytometer and inverted microscope. The crude extraction of the ginger. She put a pan and baked in at over 80 degrees Celsius, then dried the samples, were grinded with a pestle and mortar. Then she used 100 milliliters and 90% ethanol to extract the 10 grams of ginger for the 24 hours on a stir and medium speed. For 24 hours, the extract was filtered and ready to use. The cell treatment and the cell viability. The first 300,000 cells were seated in the 6-well plates. The cells were allowed to attach and get used for an environment. Yeah, frozen. So pretty much where her research transition to mine is where we introduced the mathematical modeling. So here are the ordinary differential equations that were the base for our models. So you see the exponential, the Mendelssohn, the logistic, the surface, the Gompertz, and the Bernoulli. The exponential model was often used in early growth curves and appears to work quite well in predicting the early growth. The Mendelssohn model, growth is proportional to some power b of the population. The logistic model describes the growth of a population that is limited by carrying a capacity b. The linear model assumes initial exponential growth that changes to growth that is the constant. The surface model assumes only a thin layer of cells at the surface of the tumor are dividing while the cells inside the solid tumors do not reproduce. The Gompertz model shows to provide the best fit for breast and lung cancer. In the Bernoulli model, this model shows to provide the best description for human tumor growth. And these models were used to predict long-term treatments and hopefully in the future to make them personalize these models. And for my models, I think some of the other members used methods, but for mine, a lot of the data just fit perfectly with the models. So I wasn't able, or I didn't have to use a method. I just used the equations which you see right there to the left. So in order to find which was the best fit for the data I got from Mrs. Griggs, I had to pretty much just test out all of them and just see which one best fit. So on the left, you have the exponential model, and underneath is the equation, which is v equals a, the alpha times the n, and the alpha being the 0.5, and the SSE was numbered below. So that was that. And on the right, you have the Mendelssohn model, and which was if you look at it, the optimized, it was the second closest. But as you see, the SSE was pretty big still. So that one was not a model that I would use, or wasn't the best model, wise. And then the logistic is on the left, which is the equation right now below it, which is the v equals the alpha times the n times the 1 minus the n over the beta, which alpha being the 0.5 and the beta being that number underneath, I think it's 10 million, if I'm not mistaken. or yeah, 100 million. And there was no gamma needed for this because it's not an equation. And the SSE was this number below. The amount on the right was the linear, which is the V equals the alpha times the N over the N plus the beta. The alpha being the 20,000 and the beta being 5,000. And the SSE was that number, which is really high still. And as you could tell, it wasn't the optimized, the model line and the initial guess is not really like fitting the experimental data. And the model on the left was the surface, which is the V equals the alpha times the N divided by the N plus the beta time to the power of 1/3. So the alpha was 21 and the beta was negative 110. And as you can see, the SSE was still kind of big, but smaller than the others. But as you can tell, the optimized model line was still not close to the experimental data line. And the Gompert, which is V equals alpha times N times the natural log of B over N plus gamma, which is the C. So in this model, the alpha was 20, beta was 4,550, and gamma was 50,000. And the number, that's the C number, is still pretty large, and you can tell how the optimized model line was close, or not close, but it was almost like fitting the experimental data, but the initial guess was way off. So, and for the model type, this is the Bernoulli, which was the best fit because both the optimized and the initial guess was the closest, and it fit the data the best. And this also had the smallest SSE. And the equation for this was V equals A times N to the power of 2/3 minus beta times N. In this case, the alpha was 3.5, beta was negative 0.4, and there was no gamma. And the SSE was that number right there, which is 392,377,214. So, and if you remember, on this model, pretty much is the best description for tumor growth, and all these models are the growth curves of the MCF7 untreated cells. So this is without the treatment. This is just the cells by itself. And this is the tumor, the best fit. So the results. When reviewing the data collected by the 7 ordinary differential equations, exponential, Mendelssohn, logistic, linear, surface, Gompert's, and the Bernoulli, there were several who just fit the growth curve on the untreated cells. However, the final SSE collected by the Mendelssohn and the Bernoulli seem to have the smallest and significantly close to one another. And according to the Murphy from differential equations predictions of ODE models of tumor growth are a cautionary example. He elaborates on the fact that the Bernoulli model is the best described for human tumor growth. So once we added the treatment to the equation for the untreated cells, we got this. This was the data for the dosage of the 0.11, and as you can see, the SSE, the models, the Bernoulli, I think it still is the smallest. Yeah, I think the Bernoulli and the Exponential were the smallest ones still. So in order to add the treatment to the existing equations, you had to add your given equation minus the alpha times the y. So below shows pretty much the information from Mr. Gregg's table of the dosage with all 7 models. From after retrieving each of the SSEs from the untreated cells to tumor growth and the data presented below, you're able to determine the best optimized model for the data. The burden-length model presents the smallest at 72,362,947. As previously described, this model is best utilized in human tumor growth. And here are the photos that went with that. So this was on the top left, you had the exponential, which was the graph right there. On the x-axis, you had the days, and then the y-is the number of treated cells. And you could tell right there how it came close, but it wasn't quite fitting. The same for the medicine, which is the top right. The linear, which is pretty close, but on the SSE, it didn't really, it was the highest compared to the others, but it was one of the pretty best fits for the data, like graph-wise. And on the logistic, it was the model on the bottom left. The surface model is on the top right, and you can tell how it was close, like fitting wise, but the SSE was really still pretty large compared to the others. And the Gompertz as well, but the best fit again was the burden empty, and you can see how it was the closest. Looking to the experimental data, but it also had the smallest SSE at 72,362,947. So another dosage I tried was the 0.016, which is the lowest, and the one I just showed you was the highest at 0.11. So this is the next data I got from Mrs. Griggs, which represents the mathematical dosage of the lowest from the data gathered, which was us to compare the height, see if it would change the models, and we can compare to see if burden F is still the best one to use or not. But however, this, as you can see, the burden F was not the best one to use. It was the Gompertz, but that's not it's not to be surprised, because the Gompertz is the best one for breast and lung cancer to model. So here's the graphs for the data presented right here. On the top left, you have the Exponential, which kind of really didn't fit the data, but it still was like an average SSE for the lowest. The Mendelssohn was really off the graph-wise, but it was also right next to the Exponential. It's kind of like close to the Gompertz, but it was still pretty high. The Logistic was not close in graph-wise, and it was really high. And the linear was closer, but it was still the same thing. As you see the surface, it really didn't fit the graph, but the SSE was still pretty high. And the burner and feed this time had a really high SSE, and it didn't fit the graph. And the Gompertz was the one who fit both the graph and the data on the SSE. So conclusion, therefore the results displayed the model that best fits the data collected was the Bernstein model and the Gompertz, which I did not add, but those two are the closest, because on the growth curve and the highest doses and the lowest doses, those two are all, the Bernstein was the highest in the growth curve, and in the highest dosage and the Gompers was up there in the not the second but the third and the lowest, and it best fit the data the best in the highest dosage and the growth curve. And for the lowest dosage, the Gompers fit the best, and the burn FE was really high. So those two would probably be the best to use for this cell. Questions?
You know, I have one question. I know you're a mathematician, you know, and not a chemist or a biologist, but when you treated your cells, the ginger, do you know what the protein or what the actual treatment was from the ginger? When you extracted the ginger, what actually caused the growth or the inhibition of growth? You know? I don't know. I forgot. So I'm really asking.
On the slide, I think I didn't get what she used. I just got the procedure, and I think it was the Western blot analysis. That was what she used.
Dr. Bouillon, do you remember? With the ginger? What is the active ingredient, the active, the actual activation factor there?
Oh, sorry, it was in mute instead. Okay, yeah, that actually, you know, the last summer we did a lot of Western blot.
Right, I understand, but I was trying to figure out the ginger, you know, that was crude. It was a quercetin for the onion. What is it? Do you remember what it is for the ginger?
Oh, that the ginger is like ingredient is, I think it is a ginger.
Oh, that's okay. I'll look it up. I just stopped maybe.
Yeah,
and
yeah.
One more question is I'm sure you mentioned it, but what represents the alpha and the beta? What in your equations, Andrew, what does that represent?
For each graph, it was
right.
pertaining to the graph.
Right. What is the alpha and the beta? What is that representative of?
It was just the alpha was the growth rate of the tumor, and the beta was the population.
Okay, thanks. This is very good presentation. Very good. Interesting hearing the math side of it. So that was very good.
Yeah.
On the protein, I really didn't. All I got was the cell culture, the treatment, and the viability.
Right. I understand.
Mainly, we just look like the signaling protein, some of the phosphorylated protein, how actually affected. You know, like
we just don't know which one it was.
Yeah, is the MAP kinase actually. Okay. MAP kinase is inhibited with this. It actually the bioactive compound. It is called the gingerol. Okay, that actually inhibit the phosphorylated enzyme kinase. Yeah, and also we have seen caspase that involved in the the cell killing. So that actually they increase. Okay.
Thank you so much. It was excellent. I really enjoyed that presentation. Thank you.
Thank you, Andrew. Any other question here? One of like the chat question you can see, Andrew.
I'm trying to get to the chat button.
I think the Miss Ground chat. You cannot see this chat question. How has your research impacted on the discussion about your homeopathic practices versus traditional Western medicine for ginger-based approaches?
I can honestly, I really don't know, because all I did was the mathematical side, and I really didn't like look. I looked into it as much as I got like the information, but to like further it, I really didn't look into it because I really don't know like that part of it, like the cell culture and all, like was which I knew. I don't. The Western medicine for ginger-based approaches, I can't tell you one to save my life.
Yeah, that actually the concern our, you know, in Western medicine, the people just use lots of like the chemotherapy, radiation. They actually have the some of the side effects, right? But a compound from, I mean, the plant source, or it is called bioactive compounds, actually without any side effect can kill the cancer cell. That actually the challenge, okay? And also sometimes these bioactive compounds can enhance or increase the killing capacity of the chemo. Like sometimes the drug we use to kill but have several side effects, can kill also other cells, okay, normal cells. So that actually the practice, you know, so we are using like the, I mean the vegetable or the plant source for killing the cancer cell. And the Andrew actually the working, so how this bioactive compound more efficiently or the pathway just used to killing the cancer. Okay, the more depth knowledge. Thank you. Any other question or concern? If not, maybe we can go. Thank you, Andrew, so for your nice presentation. Thank you so much. Okay, and next presenter is a Helene or Lauren? I think whose name is like, is it Lauren?
It should be Helene.
Oh, Helene, right?
Yes.
It's me.
Yeah, you are here. Oh, okay, Jim, right? Okay, great. So Helene, so you are the junior, right, Helene?
Yes, sir.
Is a biology major. She actually worked with curcumin, and one of our previous student, you know, the owner actually worked on that owner, Odumi. So she just, Helene just worked with her data to how we can do the mathematical model to get the more precise, way to see the how actually this curcumin, and curcumin, I think you know, is actually the product of the turmeric. This is another bioactive molecule. So how actually it can kill the cancer cell. So Helene, so you can share your presentation and please just present.
Okay. Hi, good morning, everyone. My name is Elaine Jimenez Jose, and I'm about to present you about my summer research, mathematical modeling to study the effect of curcumin on MCF7 breast cancer cells. My outline abstract, introduction, materials and method, mathematical modeling, with treatment, without treatment, conclusion, and reference. My abstract. Curcumin, which is an extract from the plant curcumin ligand, have been used in the therapeutic result for the clinical oncology. Curcumin is the derivative of the turmeric spice, which is yellow vinegar root group with really shape and sheet and bring orange flesh. It is originally known to be utilized in Asian dishes, but it has been discovered to have antioxidant, anti-inflammatory, antivirus, antibacterial, and anti-cancer characteristics. Curcumin has chemopreventive and anti-tumoral activities against some aggressive and recurrent cancers. Different research have established great possibility of curcumin ability to prohibit the growth of cancer cells, especially because potentially potentially too different between normal and cancerous cells. However, the Michigan Cancer Cells 7, we're going to be using and referring to it MCF7. So if you see it, that's what it means. Human breast cancer cells were discovered 50 years ago in a 69-year-old Caucasian lady woman. Also, I believe what's going on? Sorry. Okay, carry out the first observe of the effect of the various concentration of curcumin on NCF7 cells. For 5 concentration days, we concentrate which were 0, 5, 10, 15, and 25 micrometer. However, the 90% inhibit was found in the 15 micrometer of curcumin concentration within 24 hours. But the mathematical model used the higher and lower concentration to see the different result in the final sum of square due to the error that we're going to be using simplifies it to SSE, and which model fit better with the treatment using the lower and higher concentration. Therefore, the research question include understanding how mathematical model help with the effect of curcumin on the Michigan Cancer Foundation MCF7 breast cancer cells with relative to biomolecular of the cells. Breast cancer treatment with curcumin seems to be a promising medical path in the near future. Introduction. Breast cancer is a heterozygous disease and one of the major health issues that women confront. Cancer developed and process could be influenced by numerous factors, being environment and genetical factors, some of them. Breast cancer is the most common cancer found in women worldwide. It accounts for approximately 25% of the female malignant with a high prevalence in developing countries. Breast cancer is the second leading cause of cancer-related death among females in the world. Many studies have discovered that natural bioactive compounds can inhibit the effectiveness of the chemotherapy and sometimes the side effect of the curcumin chemotherapy of the current chemotherapy agent. Therefore, curcumin have been investigated to induce cells cycle arrest and apoptosis in vivo and reduce tumor size in vitro. Also, curcumin has chemopreventive and anti-tumor activities against some aggressive and recurrent cancers. On another hand, curcumin has been used for treatment in the past for many disorders for a while. Also, recent studies have shown that curcumin, either alone or in combination with other anti-cancer agents, can effectively induce apoptosis. Apoptosis is a tiny regular process of programmed cells death, including the activity. Have you seen my video?
Mm-hmm. Yeah. Okay.
Okay, so it showed me that you want me to see my video. Okay.
I didn't see your face. You didn't see your face.
Dr. Samuel, no. Okay. Can you see me?
Mhm.
Okay. Okay, therefore, to understand the complex of biological synthesis, such as cell tissues, or even the human body, it is not sufficient to indicate and categorize and characterize the individual molecule in the system anymore. It also is necessary to obtain the understanding of the interaction between molecular and pathway. This will help even to understand the complex disease such as cancer, Alzheimer's disease, or alcoholism. Mathematical modeling allows research to investigate how complex regulatory processes are connected and how those processes might contribute to the development of the disease. The preparation for the Carcoming solution, the Carcoming power was in a generous donation from the University of Texas Health Science Center at Tyler, Texas. The amylosophy Adrift Louis USA was used to dissolve carcomine to form a solution of 50 millimeter concentration. This solution was stored at 4 degrees Celsius by Honor Dewani, who brought us the data and the knowledge of the carcoming research. So I'm using her research for mathematical modeling. Mathematical modeling. Exponential. This model was often used in the early analysis of the tumor growth curve and appeared to work quickly. Well predicting error growth. The formula was B equal to AB. Mendoza is the model of growth is proportional to some power B of the population. Logistic. This model describes the growth of population that is similar by a carry capacity of B. Linear. The linear model assumes initial exponential growth that changes the growth that is contained over time. Surface. The surface model assumes only a tiny layer of the cells at the surface of the tumor are dividing, while the cells inside the solid tumor do not respond. They are mythologically inactive. Beta life. This model assumes that growth occur proportional to surface area, but that there is also a decrease of tumor volume due to the cell's death. Gorpeth. The model is a generalization of the logistic model with a signal curve that symmetrical point inflation. So the treatment that we use with my higher concentration are over here. Those are the data that I use. So for us to get the perfect data, we had to spend a quite amount of time to see which one going to be alpha 1, alpha 2. What that doesn't give us error. And the SSE, which one would be the SSE, which will be the one who tells us which one is the best fit. So those the first one, the bottom cell, is the exponential model. And this one look like the perfect fit. But this one look like a perfect fit. That's my Mendoza model, my logistic graph, my linear surface Gompers, right? So when we're looking at those models, the one that look perfect from these four is the exponential. But when we go to the graph, when we go to the data, the one that fit better with the SSE is the surface. So with my lower treatment, there was a fine micromer treatment. We use that treatment with different numbers. And over here, my closer fit to my perfect fit is the Mendoza. But when we look at it, this is my exponential, my Mendoza, my logistic, linear, surface, and Gompers graph. But when we look at it, the best fit to the graph, it will be the surface, even though with the data, the best one will be the Mendoza. The points on the data, it means the control, the treatment. The red line, it means the optimized model, and the blue line, it means initial guess. Okay. My conclusion: data collected from the data collected from the graph below by the 7 original ordinary difference equation models, exponential, Mendoza, logistic, linear, surface, Gonser, and fertilized, that were used in collecting the smaller final SSE was the surface with a 3.967743A 7050731 E09. So that's why with the higher concentration of 25 micromer. But with the final SSE for the lower concentration was Mendoza. The start with a 0, so that was the perfect fit. But if we assume to look at this treatment or at the graph for how they look, and not for the data, the best look and the higher concentration, like I said before, would be exponential, and the lower concentration would be surface, even though the data says something else. So in this program, in this model, we had to learn how to not just by the graph, but just by the data. Acknowledgement. This work was supported by the JSURE iComMathematical to Dr. Samion and Dr. Guillen at Jarvis Christian College. I just want to say I am grateful and thankful to GAF for this opportunity, and to Dr. Guillen and to Samion and Guillen for believing on me. Thank you for your time and patience to help me with this program. Thank you to my classmate of the program for helping me and supporting me. Thank you. Any questions? Oh, the reference are right here. The first reference is honor, like I say, her is the first. We took the data from her article. So, any question?
Okay, thank you, Alimi. So it's a nice presentation. So if you have any question, so please ask.
Well, I have a question. I mean, what would be did you enjoy your work this summer?
I'm sorry?
Did you enjoy your work this summer?
Yes, I did. Like when I was doing the modeling, it was amazing because it's like my first time doing the research. But also the Python is kind of difficult to do it. So we had to spend like a lot of time on Python. Plus we had to deal with if your computer is working, if it's not working. what we're doing that somebody's not doing. I have different graphs than everybody else, and my graphs are really big. So my numbers had to be different than everybody else. Everybody else is different, but my number was like really tough to work and make the graph fit. So it was cool.
Yeah, thanks for sharing that challenge. What would you think would be your next steps if you were to expand your research?
Well, I would say that I would go more deep on it, and I would try what the research is telling me, how much it grows, take that knowledge and put it in the lab. Because we already had the mathematical model. If I feel like if we put it in the lab, we can get a good result about it.
Very good. Excellent. Really good presentation. Thank you.
Thank you.
Okay, Helimi, so can you just check in the chat box, and you have one question.
Okay, just give me a second.
It is, now that your research reinforces the best models to use, what are some takeaways for treatment options?
I'm sorry, I'm reading it.
Did you see the chat?
Research reinforces the best model to use. What are some takeaways from the treatment option?
So which model you think is the best? Already mentioned, right?
Yeah.
So the option for the treatment using the curcumin. So you want to say anything?
Well, to me, that's the best model. The best model for the one that I liked the most was the higher concentration. My best model was surface, but I was going with my expectation. They would be Vertalife, the last one, but it gave me a surprise. That was the last one. It was surface. So you go with the expectation to know which model would be best for what you read, because that's another thing you have to research, not just do it, but you also have to read and research a lot of this thing to know and to understand. So for what you read and comprehend for what you're reading, your best ones are like Exponential, Gospel, or Bertolfo. But yeah, my best one is Surface and Mendoza.
Okay, any other question or anything? Okay, thank you, Helimi. So
thank you everyone for your patience. Bye.
Okay, the next is Lauren. So Lauren, so you work with garlic, right?
Yes.
Yeah, so please just introduce yourself and also open the video. You know, like we want to see you, like at least like some of the will know. So we know, but maybe other faculty or staff or maybe some students don't know so who you are. So just open the video and introduce yourself and also present your research.
Okay. Good afternoon. My name is Lauren Johnson. I'm a junior at Jarvis, and I'm gonna present to y'all what I did this summer for research.
Great.
Okay. My title is Mathematical Models of Anti-Cancer Effects of Crude Garlic Extract Compounds on MCF7 Human Breast Cancer Cells. And my research mentors, Dr. Semiono and Dr. Bouillon. So in this presentation, I will be doing the introduction, the research abstract, what is mathematical modeling, what is Python, previous research overview, materials and methods, math models, research, conclusion, references, and acknowledgments. Introduction. Abstract. The goal of this study is to experimentally and computationally evaluate the effects of crude garlic extract and to see the potential inhibitory effects on the proteins and DNA of the MCF7 human breast cancer cells due to mathematical modeling. The expected results showed that the viability of the MCF7 cells were decreased by 65% after 72 hours with the treatment at 0.1 micrograms over milliliters. The treatment showed greater inhibitory effects the larger the concentrations of crude garlic extract used to treat the MCF7 cells. 7 ODE mathematical models are introduced, which are exponential, Mendelssohn, logistic, linear, surface, Gompertz, and Bertalanffy. Using these models, I will be able to see the effects of the crude garlic extract on human breast cancer cells and determine which model represents my data the best. Introduction. Breast cancer is the most common non-skin cancer among American women. Breast cancer accounts for 15% of all new cancer diagnoses and 7% of all cancer deaths each year. Considering that 1 in 8 women in the United States will develop breast cancer in the course of her lifetime, dietary manipulation could have major impact on the incidence of breast cancer. MCF7 cells are breast cancer cells that originated from Sister Catherine Frances Malone. The name of the cells comes from the Michigan Cancer Foundation and represents Soul's 7th attempt at making a cancer line. Garlic, which scientific name is Allium sativum, a widely used spice that has been suggested as an anti-cancer agent for several decades. This herb is used throughout Asia for many purposes. When one cuts into garlic, it releases the strong odor. The odor is from the chemicals combined together with allicin. It is the component of the garlic that has the antibacterial effects. Allicin is the chemical released when the garlic has been crushed. Fresh garlic contains an enzyme called allinase and allin, which are contained in different parts of garlic plant. This unique structure is designed as a defense mechanism against microbial pathogens of the soul, allicin facts. This spice contains chemical compounds that have shown an inhibitory effect against cancer cells. In this study, the MCF7 cell line will be used to carry out the procedures and study the effects of the crude garlic extract. Mathematical models were used to determine which treatment worked best by looking at which method had the smallest final SSE. I used FOPDT modeling with the optimization method and the POW method to graph my control and treatment data for all 7 ODE models. Using these models, I found which ODE model is better for my data. This is determined by final SSE objectives, which I received from using different variables and initial guesses. Okay, what is mathematical modeling? Mathematical models allows us to quickly search and identify the most effective drug combinations for cancer patients. They are also deepening our understanding of how and why cancer cells often become resistant to chemotherapy drugs. Mathematical modeling can be used to study these types of questions, offering the cancer biologists and clinical oncologists powerful new tools to add to their arsenal in laboratory and clinical approaches. What is Python? Python is an interpreted high-level general purpose programming language. Okay, and this is the overview of the previous research given to me by Ms. Caldwell. Garlic. So the cell viability and the treatment of cells of crude garlic extract. So crude garlic extract is defined as the raw and unprocessed form of the material. This basically means the garlic has not been processed in any type of way and is used in its natural state. None of the chemicals are separated in the extract either. The cell culture is a step in which the cells begin to become used to a new environment. Cell splitting is a process in which the cells are too confluent and are split in half or an amount needed for them to grow. Plating involved using a 6-well plate or 96-well plate and adding the correct density of cells to start within each well. The garlic was freshly brought from a local market and chopped finely, then was mixed with RPMI and filtered twice to ensure no large particles were left in the mixture. The concentration of the mixture was 333 milligrams over milliliters stock, then was diluted to a 33.3 milligrams over milliliters concentration. To investigate the effects of crude garlic extract, the MCF7 cells were grown in RPMI medium with 10% FBS and 1% antibiotics and 5% CO2 incubator at 37 degrees Celsius. The cells were treated with different concentrations of garlic extract of 0.1, 0.3, 0.5, and 1 milligram over milliliter for consecutive 4 days. The results showed that the viability of MCF7 cells were decreased by 65% after 72 hours with the treatment of 0.1 microgram over milliliter. Cell culture. Once the different treatments were finished, the cells were harvested at 20 degrees Celsius in the freezer. The cells were thawed to proceed with protein extraction and quantitation by BCA method at 562 nm using ScanNet software. Results indicated that the protein concentration decreased with higher concentration of treatment. The Western blot analysis of P44/42 MAP kinase showed the inhibition of phosphorylation with increasing concentration of garlic extract. The cells were also collected for DNA fragmentation assay. The DNA analysis showed that as the garlic extract concentrations increased, the DNA in the MCF7 cells became more fragmented. The 0.5 milligrams over milliliter dosage of garlic extract inhibited the proliferation of the cancer cells and fragmented the DNA. The conclusion drawn from the study of crude garlic extract on MCF7 cells is that the allicin and chemicals within have an inhibitory effect on the MCF7 cancer cells proliferation. Okay, and these were the results for the control data and the concentrations. Materials and methods. So the materials I used for this research was Python, Spider, mathematical models, and the cell line. And these are the mathematical models. This is on the left is the one without treatment, and on the right is the one with treatment. And the brief description of each model is that the exponential model is a natural description of the early stages of cancer growth. The Mendelssohn is a generalization of the exponential growth. It was introduced by the Mendelssohn. In this model, growth is proportional to some power B of the population. The logistic model, the logistic equation assumes that the growth rate decreases linearly with size until it equals 0 at a carrying capacity. The linear model assumes initial exponential growth that changes to growth that is constant over time. This model was used in the early research to analyze growth of cancer cells. The surface model assumes only a thin layer of cells at the surface of the tumor. The Gompertz model is a generalization of the logistic model with the sigmoidal curve that is symmetrical with the point of infection. The curve was eventually applied to model growth and size of entire organisms, and more recently was shown to provide the best fits for breast and lung cancer. And then the Bertalanffy equation was created by Ludwig Bertalanffy as a model for organisms growth. This model was shown to provide the best description of human tumor growth. Okay. And this was the control graph from Python, my data results. So these are the the final SSEs, and yeah, and this is for the concentration of 1 milligram over milliliter using the treatment. And these are the graphs for, these are the results I received from Python. And these are the results for the 0.1 milligram over milliliter using the treatment. Oh, also with the control data, I did not use the treatment. Okay, and so the results were, the models are used to help us find the smallest final SSE, which is the sum of squares due to error. The results concluded that the best mathematical model for the untreated control group was the Mendelssohn model. This model gave me a final SSE of 716,470,430.7, which was the lowest of all 7 models graphed. The results collected from using the Powell method, which is useful for calculating the local minimum of continuous but complex function, especially one without an underlying mathematical definition, because it is not necessary to take derivatives. The graphs revealed that the best mathematical model used for the treatment with a concentration of 1 milligram per milliliter was the Bertalanffy model. This model gave me a final SSE of 1.115, which was the lowest of all 7 models. And the graph from using the Powell method revealed that the best mathematical model used for the treatment with a concentration of 0.1 milligrams per milliliters was the Bertalanffy model. This model gave me the lowest final SSE of 1.078. And these are my graphs. So this is the control data, and the best graph appears to be the exponential, but it was actually the Mendelssohn model that gave me the best data at the lowest final SSE. And then for the 1 milligram over milliliter concentration, the best model was the Bertalanffy, but the surface models appears the best. And then for the 0.1 milligrams over milliliters, these are both, with both concentrations I used the treatment, and the best model I got from this one was the Bertalanffy. Conclusion. In order to use these 7 ODE models for my treatment graphs, I added minus Km4 times Y to each equation, which is the number of cells multiplied by the treatment. This was applied to both my largest concentration and my smallest concentration to get the best results. To conclude, the results presented the model the best fits my data would be the Bertalanffy model, as it provided me with the smallest SSE for both of my treatment graphs. My references and the acknowledgments. I would like to thank the National Science Foundation for this opportunity. I give thanks to Dr. Semiono and Dr. Bouillon. And finally, I thank my research partners in this program and the Jarvis Christian College family. Questions?
Okay, thank you, Lauren. It's a nice presentation. So if you have any question, please.
Hi, Lauren. Very good presentation. Fantastic. It's like you guys have really been busy this summer. I just have one quick question. You know, at the very beginning, you do all your research. After you had researched all the different models that you have presented, initially, which one did you think would be the best fit? Like if you were going to have a hypothesis or a research question, initially, which would you think would be the best fit for the data that you had?
I thought it would have been the linear model.
Why was
because
that?
The equation is the most simple. And so I figured that it would be that one because I wouldn't have to put as much information into it. So yeah.
So what would you like to do next with this program, with your research?
I don't think that I will continue with the research because I do want to pursue nursing. But I think this was a great experience for me. And I got a new insight into like the biology part of nursing. So that was great.
Thank you. Yeah, it was a good presentation. Thank you.
Thank you. And in the chat box, one of the question, Lauren, if the modeling shows like the increasing resistance of the cancer cell to garlic, what might we conclude about garlic-based treatments? Just that, okay?
Mm-hmm. Resistance got you know garlic-based treatments. If modeling shows increasing.
Yeah.
He's saying that
if the model shows it increasing the resistance, right? You understand the resistance means, that means if the garlic extract not enough to kill the cancer cell, right? So in that sense, what actually your suggestion, like what actually you're gonna do?
I mean, I think it is the type of garlic. I don't know too much about the previous research, but the type of garlic we used, it might have been that type of garlic that that showed the increase in resistance. But
yeah. Okay, that is one of the like the thing we use, like garlic. You know, it containing the bioactive compound, the allicin, right? So can kill, but what actually it works, like sometimes it is used with conjunction with like the chemotherapy, like the drug we use, but that time the garlic can increase the capacity to killing. Okay, so that is one of the way we can better, I mean, get the way to kill the cancer cell. So if like, you know, like this is the bioactive molecules, we cannot say like 100% will kill, okay? But in conjunction with the chemo or drug, so we can get the more efficient killing of the cancer cell. That is the way. So if that is one of the purpose of using these bioactive compounds. Any other question? Thank you, Lauren. It's a nice presentation.
Thank you, Samiono. Oh, Dr. Samiono. Thank you, Dr. Bouillon.
Thank you.
Did you get all the questions from the chat box?
Oh, yeah.
Yes.
Only the one question, actually. Yeah, so there is no more question here. Okay. Okay, thank you. Our next presenter, Skylar, right?
Yes.
Yeah, Skylar. Actually, Skylar will present the gold nanoparticles, right?
Yes.
And it's one of the, I mean, the interesting topic for Dr. Carter. Okay, actually, Dr. Carter involved so much like the nanoparticle work and helping our students. We're really grateful, Dr. Carter. So we just, Skylar actually used the honor, right? the data to model mathematically. So, okay, Skylar, just open your video. Just our audience want to see you, and then you share your presentation, and please present. Thank you.
Okay, well, this is the final presentation that I worked on week 7. During week 5, I was done with the control group. So basically, this is an extension of week 5 since I finished with the rest of the groups. So today we're going to talk about my research paper, and it was using modeling mathematics to study the effects of gold nanoparticles on human MCF7 breast cancer cells. So the abstract. There is no cure for cancer, but research has proven that some targeted treatments can increase the survival rate of the patient. The problem is, what doses are most efficient to increase survival rates in these patients? This study is focused on using mathematical models to study the effects of gold nanoparticles on Michigan Cancer Foundation 7 human breast cancer cells. Based on the data and laboratory experiments done by Honor Adewemi, I will be answering the question, which ODE models is most efficient in data used in targeted treatment options? Ordinary differential equations models are a set of differential equations involving functions of only one independent variable and one or more of their derivatives with respect to that variable. Cancer is a general term for diseases that is controlled by growth of the abnormal cells that can happen anywhere in the body. Some cancers, like breast cancer, can be genetic. Based on data collected and literatures on mathematical modeling, I was able to compile each model in Murphy's paper, Differences in Predictions of ODE models of tumor growth in Python. With the smallest sum of squared due to error or least square regression in mind, the Bertalanffy model fits almost seamlessly with the data provided. The results indicate that the Bertalanffy model, which is best to describe human tumor growth, best fits this data. MCF7 breast cancer was proposed by Dr. Herbert Soll and was the first cell line to be hormone responsive. Cancer is a general term for diseases that is controlled by the growth of abnormal cells, can happen anywhere in the body. These cells accumulate and can form tumors. Even some cancers, such as breast cancer, can be genetic. As mentioned before, there is no cure for cancer, but there are a handful of treatments. In science, mathematical models have been used to explain how a system behaves. Mathematical models can be dynamic systems, statistical models, or game theoretical models. They have been used to determine the best treatments and schedules for increasing survival rates. On the other hand, nanoparticles, such as gold, have been used for treatments in the past for many disorders for a while. Gold nanoparticles are used for many research topics to be synthesized for treatments on diseases and many other alternative medicines laboratories around the world have been researching on. Gold nanoparticles have been used on various medical treatments over periods of time without any harmful effects. When gold nanoparticles reach a diameter of 5 nanometers or less, they can be used as a catalyst. Nanotechnology has been used in various different types of ways and treatments for research using nonmetals and metal particles that are going to target cancer cells and tumors. The advantage of gold nanoparticles is it will specifically target the organelles and the tumors. Gold nanoparticles can be changed in their interaction with external systems. In this study, I will be using mathematical modeling to analyze the effects of gold nanoparticles treatments in MCF7 breast cancer cells based on data and laboratory experience done by Honor Adewemi while in the undergraduate research program. So the materials and methods I used were cell culture, synthesis of gold nanoparticles, treatment of cells, cell viability by MTS assay, mathematical model methods. So when doing the cell culture, cells were purchased from American Type Culture Collection and maintained in 5% DMSO at -80°C until use. For culture, cells were maintained in RPMI 1460 medium, enriched with 10% fetal bovine serum and 1% antimycotic, at 37°C with 5% CO2 in humidified atmosphere. Cells were plated at 5 × 10⁴ cells per well to obtain a pattern of growth for further studies. The nanoparticles were synthesized using a modification of the Turkovich method by Sesbron et al. Briefly, 0.01 gram of hydrochloric acid. Sorry. Why can't I? I'm so sorry. Was added to 50 milliliters of heated water. 1% of sodium citrate was then added to the heated stirring solution. A reduction reaction was noticed when the solution turned into a wine color. The solution was purified using a 0.2 micrometer filter, protected from light and stored at 4°C. MCF7 cells were seated at 3 × 10⁵ cells per plate, allowed to attach for 24 hours, and incubated in a humidified atmosphere at 37°C with 5% CO2. The cells were treated with 0, 5, 10, 15, and 20 micrograms per milliliter of gold nanoparticles in cultured media. Cells were counted using the improved Neubert hemocytometer chamber for 5 consecutive days. The MCF7 cells were plated at a density of 1 × 10⁴ cells per well in 96 well plate and allowed to attach. Thereafter, the cells were treated with gold nanoparticles at 0, 5, 10, 15, and 20 micrograms per milliliter for 72 hours. Manufacturer's instructions were followed. Briefly, 20 microliters of MTS solution was added to each well and were incubated for 4 hours. These are the mathematical models I used in this research. In order to find out what mathematical model would be best to describe the data, I had to try 7 different ODE models. The models and equations are the exponential model, Mendelssohn, logistic, linear, surface, Gompertz, and Bertalanffy. And these are the equations that were used in the research. The results. The next model with the smallest SSE would be the Bertalanffy model and fits almost seamlessly with my data. The Bertalanffy model is used to describe human tumor growth, and it fits really well. These are all models and SSEs for the control group. And you can see the Bertalanffy model has the smallest SSE. And next I'm going to talk about the models with treatment. When adding treatment to the equation, you must subtract the number of cells multiplied by the treatment. These next mathematical models will show the effect of 20 micrograms per milliliter dose of treatment on the cells throughout 5 days. Keep in mind that with this dose, the optimized model may not be the same as the model for the growth of the tumor cells. The Bertalanffy model. This model fits almost perfectly with the data used in this research. As stated before, the Bertalanffy model is used to describe human tumor growth. When using the Nelder-Mead method, the final SSE output is 1,063,000 163,071,771.317291. Oh, I skipped one. Other models used and their SSEs. And I put them together not in order to their SSE, but the closest SSE and how the models look almost alike. So the exponential model and the Mendelssohn model have almost similar SSEs, and the optimized model fits okay with the research. This is the Logistic and Bertalanffy. They also, well, they don't have similar SSEs, but I put them together because they almost look the same. These are the Gompertz and Surface. For some reason, I couldn't really get them to follow the data because I would always get like a step error, or there's too big of a number used, or it just didn't make sense with the equation. But they are similar in final SSEs and in shape. These next mathematical models will show the effect of 0 micrograms per milliliter dose of treatment on the control group throughout 5 days. The control group is solely to contrast the treatment group. The Mendelssohn model. In the control group, the Mendelssohn model best described the data. In this model, growth is proportional to B of the population. The Nelder-Mead method was also used. Optimized model fits, but initial guess does not. So the initial guess is the blue line, and the optimized model is the red. The final SSE was around 1 million. And these I'm gonna show you the other models used and their SSEs similar to before. This is the exponential and surface model. They both have an SSE around 13,000. They look almost identical, but you can see that on day 4, it fits a little better on the surface than the exponential. The linear and logistic model also look alike with an SSE of around 33,000 for the linear, and the logistic has one around 96,000. They look alike except that on day 4, on the logistic, it kind of goes straight. And that, I guess that explains why the SSE is not as similar to the linear. The Gompertz and the Bertalanffy model, they look almost exactly the same, and they do have similar SSEs, but the Bertalanffy has the smallest one from the Gompertz. The results indicate that the Bertalanffy model best fits for data collected on the tumor cell growth. It has the smallest SSE and fits the data best. In Murphy's paper, the author elaborates what each model is used for. Limits in the research include not using chemotherapy, although using data from existing treatments could prove useful to find the solution to a more useful targeted chemotherapy. And those are my references. Is there any questions?
Okay, thank you, Skylar. So it's a nice presentation. So any question? Any question? So Skylar, what actually your explanation like about the mathematical modeling to I mean in prospect of the cancer killing?
Well, with the what medium did she use? I think she used, let me see. Right here, I think she used, oh, she used sodium citrate. I guess that has a pretty, how do you say it? A good hindrance to the tumor cell growth. And that's why whenever you see the difference between how the cells actually degenerated with this sodium nitrate compared to no, not using any.
Yeah, so now what actually the explanation here? So is a good fit model, mathematical model, you can precisely say like something, what actually happening inside the cell?
Well, with the Bertalanffy model, no, that's.
Which one is the good fit here?
That's the control group. Let me see, hold on.
Treatment.
So with the models of treatment, it was the Bertalanffy model. It is used to describe the tumor growth, but in this case, it's not, it's regressing. So that's, I guess that's how I'd explain it. I mean.
Okay. And one in the chat box, Miss Graham, you see it's a very good question actually. It's a very nice question. Once the most effective models are identified, what are the next steps in your research using this model or in your results? What actually you next plan, what you'll do in your...
Okay, well, since the Bertalanffy model is describing how the cells were not growing, you would try and find a treatment that could possibly inhibit the receptors, and then I guess you can try and do another model based on that research.
Okay. Any other question or anything? Okay, thank you, Skylar. It's a nice presentation. Any question? Okay, great. Thank you. Okay, the next and not the least, I think the Megan is the last presenter. So please open your video and then introduce yourself and sharing the presentation, please. Thank you, Skylar. The Megan, please.
Yeah, no problem. Hold on.
So can you just open the video if possible? Then our audience can see who actually is the Megan. You don't have any option or?
No, it's not open yet.
Okay.
I saw you.
Oh, okay, okay. Sorry. Okay, yeah, it's good. Thank you. Yeah, you can start, please.
My name is Megan Ibarra, and I am going to be presenting the mathematical modeling to study the effects of synthesized palladium nanoparticles using Gardenia jasmoid endless fruit on adenocarcinoma cervical HeLa cells. The outline is going to be the abstract, the introduction, what is mathematical modeling, what is Python programming, materials and methods, mathematical modeling of palladium nanoparticles, treatment results, growth curve model of HeLa cells, treatment models of a dosage of 0.88 nano. Control models, results, conclusion, and references. Abstract. Cervical cancer is a term, the silent kill, due to the fact that symptoms do not appear later until its later stages. By this time, the patient will need a stronger and targeted chemotherapy treatment. Study shows that palladium nanoparticles prove to be a viable option in cancer treatment. Therefore, in this study, we are going to further the study on mathematical modeling of synthesized palladium nanoparticles using gardenia jasmine Ellis fruit on adenocarcinoma cervical HeLa cells. Abstract. Continued. What is mathematical modeling capable of describing tumor growth in the absence of chemotherapy, and what is chemotherapy? Present in this definition, a mathematical model is a model that uses mathematical language to describe how a system behaves. In this case, the models we would describe on how cancer cells grow and how they are affected by the palladium nanoparticles. This study utilizes a cell culture to grow cells and viability to manual count the cells, jasmine fruit extract to synthesize palladium nanoparticles, and extraction method, and spider to work on mathematical models. After analyzing each model, the surface model has data provided best. Introduction. Cancer is an abnormal cell growth and uncontrolled cell that can invade and infect the surrounding healthy tissue. Cancer can be very aggressive to the human body, meaning cancer destroys healthy tissue that we have in our body. In this study, we are going to be talking about cervical cancer, but from the HeLa cell line. HeLa cells originated from Henrietta Lacks, who was diagnosed with cervical cancer but later died from aggressive form of cancer. Cervical cancer is a type of cancer that occurs in the cells of the cervix of the lower part of the uterus that connects to the vagina. HeLa cells are a model cell line used in a scientific research. HeLa cells are the oldest and most commonly used in the cell line. The cells originated from Johns Hopkins Hospital, where she had gone to get her treatment, discovered by Dr. Gregory Gay, who was researching methods of immortalizing cells long enough to experiment with in vitro. With that being said, metal nanostructure are gaining an interest in a lot of biomedical applications owing their shape and size, electronic, mechanical, and optical properties, and their outstanding resistance to corrosion and oxidation. Metal nanostructure has offered many possibilities for cancer therapy, targeted drug delivery, and diagnosis. Palladium nanoparticles have been used on various medical treatments over time without any harmful effect. Palladium can be used as a catalyst for hydrogenation by acting as an absorbent material. Nanoparticles and the use of nanotechnology now play a major role in biomedical research. Recently, nanoparticles have been used to targeted drug delivery, cancer immunotherapy, and help with diseases, diagnosis, and treatment. In this study, I'm going to further experiment in mathematical modeling to study the effects of palladium nanoparticles using gardenia jasmine Ellis fruit on adenocarcinoma cervical HeLa cells. What is mathematical modeling? Mathematical modeling is a description of a system using mathematical concepts and language. The process of developing mathematical is termed mathematical modeling. Mathematical models are used for natural sciences such as physics, biology, earth science, chemistry, and engineering disciplines such as computer science, electrical engineering, and as well as social sciences such as economics, psychology, sociology, and political science. What is Python programming? It's a general purpose programming language designed to be used for wide variety application. It supports multiple programming and objective-oriented and functional programming. Python defines the type of objects to build into your code. Unlike some other languages, you don't need to declare the object type. A huge number of object types of functions and method are available for use without importing any external modules. These math functions list calls to computer systems. Explanation of palladium nanoparticles. Palladium nanoparticles have been used on medical treatments over periods of time without any harmful effects. Palladium nanoparticles and much attention in the have much attention in the field of biomedical applications due to their huge catalyst and sensor activities. Palladium is a rare earth metal with potential biomedical treatment opportunities and advances in nanotechnology. Gardenia jasmine ellis fruit is known for disease treating available. Gardenia jasmine ellis fruit is said to treat anxiety, agitation, bladder infection, bleeding, cancer, constipation, depression, diabetes, and many other disorders. There isn't much information about the remedies made with the fruit or if they truly work. Materials and methods. The HeLa cells, palladium nanoparticles, Gardenia jasmine ellis fruit, and the Python modeling. Methods. The cell culture. Adenocarcinoma cervical HeLa cells were obtained from American type culture collection and were maintained at a log phase. So the cell grows by culturing in RPIM 1640 supplemented with 10% fetal boiling serum and 1% antibiotic anthromycotic at 37 degrees Celsius and 5% CO2 air. So viability, take 10 in a growing cell and 10 microliters in an endophore Egyptian blue and an endophore tube, mix well with a gentle shake and place a small amount of cell mixture onto a hemocytometer and manually count the cells under an inverted microscope. The synthesis of palladium nanoparticles. The palladium nanoparticles were synthesized in a 125 milliliter glass bottle and were heated to 7 degrees Celsius in a temperature regulating water bath. The ratio of palladium chloride solution to the jasmine cake ellis fruit was 1 milliliter of palladium chloride solution to 24 of Cape jasmine fruit extract. The nanoparticles were synthesized for 24 hours at 70 degrees through a filter of two filters, then covered the aluminum foil, then refrigerated until needed for treatment. The extraction of the Cape jasmine ellis, the fruit extraction method. Put 2 grams of blended dried fruit of 10 milliliters in deionized water, stir and heat for 5 to 10 minutes and filter until the fruit extract is clear of any debris. The mathematical modeling equations were used was Mendelssohn is a generalization of exponential growth model was introduced by Mendelssohn. In this model, growth is proportional to some power b of population. Logistic, this model describes the growth of a population that is limited by carrying capacity of b. The logistic equation assumes growth rate decreases linearly with the size until it equals 0 at the carrying capacity. The linear model assumes the initial exponential growth, and that changes to growth that is constant over time. The model was used in early research to analyze cancer growth of cancer cell colonies. The next one is surface. The surface model assumes only a thin layer of cells at the surface of the tumor are dividing while cells inside of the solid tumors do not reproduce. They are mitotically inactive. Gompertz, the model is a generalized of a logistic model with sigmoid curve and asymmetrical, which is the point of inflection. The curve eventually applied to the growth model of entire organs and recently was shown to provide the best fit for breast and lung cancer growth. Bertalanffy, this model assumes that growth is proportional to the surface area that is also a decrease of tumor volume due to the cell death. This model was shown to provide the best description of human tumor growth. Lastly, the exponential, the proportional constant A is the growth rate of the tumor. This model was often used in early analysis of the tumor growth curves and appears to work quite well at predicting early growth. This is the treatment results over 3 days with 3 concentrations. The growth curve of HeLa on Python as the results for the model that best fits the growth curve and had the smallest SSE. The Bertalanffy model had the best fit to the HeLa cell growth and had the smallest SSE out of the 7 models that were conducted in Spider. The Bertalanffy model is said to be the model that assumes the growth occurs proportional to the surface area, but that there is also a decrease in tumor volume cell due to the cell death. And this is the growth curve of HeLa cells over 6 days, and the SSE that was given was 2.00. This is a treatment model with a dosage of 0.88 nanometers. As we continue to do the mathematical modeling for the treatment, we started to do the same process we did with the growth curve treatments. We are still looking for the smallest SSE for the treatment model, so the best fit is going to be the surface model. The smallest SSE and the best fit for the treatment growth was the surface model. The surface model assumes only a thin layer of cells at the surface of the tumor dividing while the cells inside of the solid tumors do not reproduce. They are mitotically inactive. And the SSE given for this one was 1.346. And then this is the control model. Finally, we get to the mathematical modeling of the control data. The best fit for the control data was conducted on Spider. It's a Bertalanffy model SSE. There's also 2 more models that had the perfect fit, but the SSE was too big, which were the surface and the Gompertz. The final SSE was 1.68. My results. The results for mathematical modeling was being used. Bertalanffy was the best fit to collect the tumor growth curve. Surface plot had the smallest SSE to fit the tumor models with the dosage of 0.88 nanometers and finally the best fit and the smallest SSE for the control models is the Bertalanffy models. In conclusion, I furthered the study of studying the effects of synthesized palladium nanoparticles using gardenia jasmine as fruit on adenocarcinoma cervical HeLa cells that was conducted by a previous student using the mathematical modeling and other models that are used said that modeling can help the growth of the tumor rate size and can prevent the size of the tumor getting a certain size. The results that show that the tumor growth model can lead to as much change and predicted outcomes, and that the model that best fits the experimental data might not be model that best predicts the growth. I have gained the knowledge that we can use mathematical modeling to further cancer treatment, and is going to be very helpful to further studies to figure out more helpful and non-chemotherapy treatments. As the advancement in medicine is advancing, there will always be that mathematical modeling will be the next treatment for cancer. Acknowledgments. I would like to thank God for giving me the knowledge of the patients and the strength to accomplish this research. Next, I would like to thank the NSF for funding this research program, and Dr. Samuel and Dr. Bion for being patient with me and always pushing us to do our best in this research. Lastly, my lab mates for the constant support throughout the summer program. And
Great, thank you. It's a good presentation. Yeah, again. You did a good job. It's great. So any question? Any question? How about the Dr. Carter? Are you hearing?
Yes, I'm hearing. That was
So you
really good.
think
It was really interesting. I did have a question, and it was really interesting because this was a different cell line, the HeLa cell line that you were dealing with. So
Yeah, yeah.
I was really interested to see if you were going to actually get a best fit for your model, but it seems to be the same. So I was wondering if the treatment, if you would say the treatment or the cell line was the most important in predicting the model for this, the actual curve.
I think it was the cell line, how unique it is from other ones.
But it pretty much gave the same results, right, as the others, the MCF7, which had also different treatments. So I was just wondering if there, I guess this would probably be further studies, of course, but what would be the significance in getting pretty much the same fit?
Yeah. So that is one of the interesting way we can find out which, like, the model or like the cell line is good. Okay, yeah, is it the same cell, I mean the model, or is the different? So you can, yeah, maybe later, like in future research, you can see this. Yeah.
Good presentation, thank you.
Very good. Any question? Yeah, it's a long presentation, right? It's more than 2 hours. So I'm really thankful everybody to be here and patiently you hear our researcher, I mean the accomplishment, because they really work hard during all like the summer 8 weeks program. It's a great opportunity for the students, and also Dr. Samuel, actually, he did a, I mean, great job, right? I mean, he actually took a very, I mean, most of the time actually. So every day, and also the students also work hard. So that actually the accomplishment of the research. So now we are actually planning, and students are planning to present in the conference, national conference. Dr. Carter, you know, like the last few years, like the several years, actually, our student presenting in very famous, like annual biomedical research ever come, right? This year also the same program, and it's nice that this year actually is very close to, and it is in Texas this year, actually, San Antonio. Okay, so I believe our student will present in this conference, and that actually helps, okay, your career. Okay, and I think like the Justin was here actually today, and you know the Megan actually continue the work of Justin, because he was here today, and he is now in UT San Antonio Medical School. Okay, for
wonderful.
And he just in the beginning and he was most of the time here. I just saw him, and also I talked to him also. So he really excited, you know. So that is actually our achievement. Okay, so if you have some research background, you have the opportunity this type of program. Okay, so you can go, you can show that I have this opportunity, I have this experience actually. So yeah, Justin actually, because I don't see now, maybe he's now in lunch.
He was he had to leave. He was doing something in the lab.
Left, right? Yeah, but you know that he was actually here. So that actually the purpose of this research. So, and from the beginning, you know, so Dr. Carter actually helping our students and all the time, and she is actually a great researcher, okay, in this field. So we hope we will help.
Or you, Dr. Bouillon.
So,
You
Dr.
have really
Samir
put your time in, and we're so proud of you.
Thank you, Dr. Carter, and also Dr. Samiano. Now
And Dr. Samiano, yeah.
He's just adding new, research technology. Actually, the mathematical. Great, right?
Yes.
So I'm really happy that so we can actually continue this research, okay? And always Dr. Carter is helping us, Dr. Samiano, and we work all together. And also, you know, that we are planning for the next grant, okay? So that is the thing. So any question, anything, any concern?
No, sir.
Dr. Samiano, you want to say something?
Yeah, I'm speechless, you know. Speechless.
You just open your video.
Yeah.
Like you, speechless.
You know, actually we planned only like 5 students, but apparently there are 6 students who apply for the program, right? So, and then we saw that all of the students like good students, so we accept all of them.
Yeah, so they did a great job.
Yeah.
Okay, so and final conclude, I think Dr. Lang, so you can just finally conclude our program. So Dr. Carter, Dr. Lang, are you hearing? So yes, so our presentation already done. So if you want to just finally conclude the program.
Well, I've enjoyed myself. I can't say that I learned a lot, but that's only because I have a deficit in science. But the presenters were very informative and very enlightening, and I'm just so glad that I had an opportunity to witness all of this. And young ladies and gentlemen, you're doing some great things in this world in which we live in, and I want to encourage you to continue the research that you're doing and expound upon new research. Remain innovative because you are our future. And again, thank you for all that you have done for yourselves in Jarvis and the community in which we all live to help further our understanding of how things can be better. So continue your research. Find a cure for cancer. Thank you.
Thank you so much. Dr. Carter, any word?
I'm just so excited that we were able to continue research during this pandemic. And using the mathematical model is just great because it's no limit to how far you can go. You don't have to be limited by certain chemicals or certain cells that may be toxic to the, really, really toxic to your body as well as to the cells. So I'm really excited about us, you guys getting a hold on the mathematical model. And I'm excited about the kind of proposals that we can write and where we can go with this, because we are down for the semester in terms of having to actually have hands-on. So still we can continue the research model. So that's what I'm really excited about. So you guys, it's wonderful and really, you did a great job.
Thank you.
Thank you, Dr.
Okay, Dr. Samiano, you want to say anything? Or?
Yeah, I did, right?
Okay. Okay, thank you so much. Thank you all. You actually attend and also hear our student achievements. So thank you so much. Thank you. Okay, so. Thank you so much, all presenters.
Okay, Dr. Vian, what is the next step?
Yeah, so Okay, so class already started. So the next we have a meeting, right? Is it
Yeah, we
4
have
o'clock?
meeting, and also have class at 2:30.
Oh, 2:30? Oh,
Yeah.
yeah. So
What is it today? Yeah, today is a Thursday, so I have a class at 2:30.
2:30.
Yeah.
So,
Okay.
Yes.
so
Yes.
for the class that because as we supposed to be a ecology lab, for those, are we going to get a excuse to be late? Because the classes start from 1 to 2:50.
I think doctor, you just at 1 to 2.
2:50, I
Yeah,
think.
just join right now and say so. You had a, the research presentation, and I think the doctor will understand. Okay, don't worry. Just join right now.
Okay. Thank you guys for everything.
Yeah, yeah. Thank you.
Welcome.
Okay. Bye.
Bye-bye.
Okay. Yeah, Dr. Samiano is
I'll talk to you guys later.
okay. Thank you, Jordan.
Okay. So yeah, we need to talk about the conference, right?
Yeah.
When is the due date, Adrian?
Huh?
Oh, the SACNAS is October, so like SACNAS is earlier, so the SACNAS conference
Oh, okay.
is in October, so I don't know if it's
October?
yeah.
Okay.
But
And
it's
now,
virtual. That is virtual.
okay. It's a virtual, right? Okay.
Yeah.
Yeah, so I think
It should be in like in Long Beach, California, but they cancel. They have it like virtual.
okay.
So they don't have any face-to-face conference and all that. So they totally cancel the face-to-face and only they have the virtual conference.
Mr. Carolino, so you are here? Oh, you are here. I didn't say. Okay, great.
Yeah, he's here.
Oh, he's here. Okay.
He's here all the time.
Oh, he was all the time. Yeah. Okay, great. So, okay, Dr. Samiano, so maybe we'll talk about this. And at least a week before, maybe we have to again remind our students. So this is the time you have to submit the abstract. Most of actually they have that.
Like them take us, right?
Exactly.
Okay.
Okay. So talk to you later. Okay. Thank you.
Okay. Thank you, David.
Thank you, Mr. Carolino.